The last chapter we will cover this summer will discuss evolution and how the acceptance of evolution has changed over the years. Charles Darwin, on the left, is credited as the first scientist to theorize about evolution as a slow, gradual process. At about the same time Darwin was proposing his ideas, Alfred Russel Wallace, on the right, was also proposing a similar theory. The two men did not work together, but both traveled the world to explore and research their ideas. Although Charles Darwin is certainly the most well-known scientist credited with ideas of evolution, there were a few philosophers who came before him. A few of the scientists who had thought of evolution include Aristotle, Leonardo da Vinci, and Jean-Baptiste de Lamarck, along with ideas from James Hutton, who believed the Earth slowly changed over many years, allowed these scientists to theorize about extinct species and changes in species. In 1831, Darwin boarded the HMS Beagle and spent 5 years traveling around the southern part of the world. During his voyage, he studied and collected many species, observing similarities and differences between them. These observations were the origin of his thoughts on evolution. Darwin's idea started with the notion that organisms best adapted to the environment would survive, passing on traits to the next generation. If an organism was not suited to survive, they would perish, thereby decreasing less favorable traits in the population. The generations that followed would have a higher frequency of traits favoring survivability, and fewer, less favorable traits would be passed on. A clear example of natural selection lies in the example of iguanas found on the Galapagos Islands as compared to their ancestors, which are from the mainland of South America. Watch the linked video to see how the island iguanas adapted to survive the new environment. For natural selection to occur, variation must exist in a population. Variation can be both beneficial to a species, improving survivability, and detrimental, decreasing survivability. These variations can lead to adaptations in a species which improve survival and thereby reproductive success. If adaptations diverge enough from the parental species, a new species could be developed. In the case of the Galapagos finches, Darwin recognized many variations depending on the environment. The birds had adapted to the environment depending on the food source. Beak size and shape differed depending on what the birds had available to eat. The husband and wife team of research biologists Peter and Rosemary Grant have spent decades observing the Galapagos Island finches. In the years they have researched the birds, they have observed evolution firsthand. Watch the linked video for their story. Evolution takes time. It will not happen in a single generation, but as the Grant team demonstrated, it does not have to take thousands or millions of years to happen. In order to understand evolution, the term species is very important. A species is a group of similar organisms that can breed with each other, producing viable, fertile offspring that will sustain the species for future generations. Notice the salamander picture. When the mountains separate the two groups, they are separated long enough that when they are able to physically interact again, their genetics have changed enough that they no longer are able to interbreed. They would then be considered two different species. A population is a group of individuals that live in the same area at the same time. Think about the population of Baytown. Do you include people that live, work, or attend school in Mont Belvieu as residents of Baytown? No. Only the current residents in Baytown would be included in the Baytown population. The same is true with organisms. To consider a population of birds, only the birds of one species in a defined geographical region would be considered part of the population. When one species has enough variation, as in the reproductive parts of flowers shown here, The species can diverge enough that they no longer remain one species and begin to require different needs to survive, different environments, different insects to pollinate them, etc. This is known as divergent evolution when one species becomes two different species as a result of these changes. When very different species adapt with similar phenotypes, it is known as convergent evolution. These species remain different but have traits that are similar. For example, these three distantly related mammals adapted independently to eat ants and termites in similar grassland and forest environments, but in very different parts of the world. The aardvark, as noted in the top A picture, is a native to many parts of Africa. The giant anteater bee is native to southern Mexico to northern Argentina, while the pangolin C is native to parts of Asia. Another example of convergent evolution is when two unrelated animals adapt to an environment. For instance, the Arctic fox in the lower A picture and the ptarmigan, the lower B picture, both evolved white coverings in the winter to help hide from predators. For a quick comparison of divergent and convergent evolution, watch the linked video. Analogous features are similar in how they look and how they function, but are not from a common ancestor, while homologous, homo meaning same, share a similar appearance and function because they are derived from a common ancestor. For more examples of comparative anatomy, watch the linked video. There are two ways of looking at evolution: microevolution and macroevolution. Microevolution is the small changes that occur within a few generations that can result in a change in genetic frequencies. Macroevolution takes many generations, usually thousands if not millions of years, producing a variety of different species from a common ancestor. In the example of the moths, microevolution occurred during the Industrial Revolution when trees became soot-covered and much darker. Before this happened, light-covered moth genes were more prevalent in the moth population. However, after trees became soot-covered, the darker moths had an advantage of camouflage as compared to the lighter moths. As a result, over several generations, there were more dark-colored moths in the population as compared to the light-colored moths. This example of microevolution does not result in a new species, but simply a change in the genes that are passed to offspring within a species. For macroevolution, one species will become two or more species over many generations and thousands of years. Notice the cat evolution chart. All four cats, which are unique species, evolved from one common ancestor. For a comical take on micro versus macroevolution, watch the linked video. Adaptations and evolution within populations lead to a field of science known as population genetics, where scientists study the genetics of a population. Over time, the alleles, which remember are variations of a gene, can change as noted in the moth example in the previous slide. The gene pool of a population includes all possible alleles for a given gene. Environmental conditions can change the frequency of the alleles in the gene pool. Using the moth example, when the tree bark was lighter in color, the frequency of the light-colored allele was higher, but when trees were soot-covered, the dark-colored allele was expressed more often. The Hardy-Weinberg equilibrium is a mathematical formula used for predicting the chances of homozygous versus heterozygous offspring. This formula only works, however, if there are no environmental or evolutionary impacts causing changes in the alleles of a species. Two important aspects of evolutionary impact are mutations and genetic drift. Mutations happen randomly in the DNA sequence of a gene. Mutations can serve one of three functions in a population. They can increase survivability, they can decrease survivability, or they can have no impact on the population's survivability. Genetic drift is also a random occurrence causing a shift in allele frequency within a population. These can happen as a result of selection pressures such as finding a mate, food resources, predation, etc. This drift could even eliminate an allele from the gene pool of a species. Look at the rabbit example. In the first generation, the allele frequency, as noted above the rabbits, is equal. There are as many dominant capital A alleles in the population as there are recessive or lowercase alleles, a 50% to 50% split. If, however, only the 5 rabbits circled in red were to breed due to environmental pressures at that time, the outcome decreases the chance of a homozygous recessive allele in the resulting second generation offspring. Now, in the second generation of offspring of rabbits, the allele frequency has shifted to 70% capital or dominant compared to 30% lowercase or recessive. Then, if only the 2 rabbits in the red circle from the second generation were to mate, both of which are homozygous dominant for brown, all of the resulting offspring would be homozygous dominant, completely shifting the allele frequency again. The dominant allele would be 100% and the recessive allele would be 0% in the third generation. When unexpected natural events such as hurricanes, floods, or volcanic lava flows, or human-impacted events such as clearing a field for construction, some species are impacted when a large portion of the population is killed off. This mass killing changes the gene pool of the surviving population drastically, impacting gene frequency, and is known as the bottleneck effect. Another way that genetic makeup can shift is known as the founder effect. In this case, a small group gets separated from the original population. When the new population produces offspring, the genetic makeup will resemble the founding parents and not the original population. This is known as the founder effect. For another example of these two types of genetic drift mechanisms, watch the linked video. When an individual of a population travels or migrates to another area where a different population of the same species is, the traveler could carry different alleles into the new population and out of its original population. This can also happen with plants if the wind or an animal carries seeds to a new region. Different alleles can be introduced into a population, and this is known as gene flow. Fossils are used as evidence to support the theory of evolution. Scientists can use the fossil record to show the changes in organisms as the landscape changed over millions of years. For example, when the landscape of parts of North America were dense forests, the ancestor of the modern horse was a much smaller mammal. As the forest transitioned to prairies, the animal's body shape, feet, and legs, as well as teeth sharpness, adapted to improve survivability in the changed environment. As macroevolution can result in a wide variety of organisms originating from a common ancestor, some body parts evolve and are no longer necessary. These anatomical parts are referred to as vestigial structures. For example, although a whale has fins, the bone structure within the whale's fin is homologous to the bone structure of a human, a dog, and a bird. This indicates that these organisms share a common ancestor from millions of years ago. Over time, the need for digits in a whale were no longer necessary, but have remained as non-functional. Another example is the pelvic region in a snake. Although snakes do not have legs, they have vestigial structures including ischium, which is part of the pelvis, femur bones, and claws. This shows us snakes are descendants of reptiles that did have functional legs and claws. Embryology is another type of evolutionary evidence. When looking at widely varied organisms, similarities can be seen in the embryonic stages. Sequences within the DNA of each respective organism can turn on or off certain genes during development. Also, mutations in the sequence from one species to another can dictate which form the adult might take. Notice in the image, all four organisms have gill slits and a tail during embryonic development, but by adulthood, some of these traits disappear in some species. These similarities can be used to identify organisms which share a common ancestor. For an animated comparison of the embryonic development in humans and related animals, watch the linked video. The study of how plate tectonics move continents over millions of years is also evidence to help support evolution. With the movement of continents, some organisms become isolated from other members of species and diverge into new species, which are not found anywhere else in the world. This is especially true with species found on islands. On the other hand, when a species evolved prior to the continent spreading far and wide, these species can be found in many parts of the world, as noted in the picture with the Proteus flower found in parts of the southern hemisphere. Molecular biology has greatly impacted the ideas of evolution. Many organisms have been reclassified due to genetic similarities to otherwise very unlikely relatives, as well as reclassified due to vast differences in genetic makeup despite being physically similar to other organisms. As a result of all organisms using a genetic code that is nearly identical, evolution is further supported. An example of a species is a group of lions. They cannot successfully mate with other big cats as their genetics are varied enough that they could not produce viable, fertile offspring with other cats. These happy-faced spiders, on the other hand, although they look different, are the same species and therefore can successfully produce offspring. There are nearly 9 million species which have been identified and countless ones which have yet to be identified. But how do we end up with so many different species that can look similar to each other, like the big cats of Africa? Speciation is why these big cats of Africa cannot crossbreed. Speciation occurs when one species evolves to become different, distinct species and can no longer successfully reproduce with similar organisms. This process typically takes many generations, possibly up to millions of years, with help from a speciation event to cause a divide in a group of organisms. Notice in the left image, several finch species descended from a single ancestor. These birds became unique species due to available food on each island, changing the shape of their beaks. In the right diagram, it shows how wild dogs, foxes, coyotes, wolves, and domesticated dogs have become separate species. For a look at speciation in action, watch the linked video. The divergence of a species into two or more species results from reproductive isolation. As the name suggests, two groups of organisms become reproductively isolated. There are two main types of reproductive isolation: pre-zygotic mechanisms, which prevent a viable zygote from ever forming, and the other is post-zygotic mechanisms, which occur after fertilization but produce non-viable or sterile offspring. In the left picture, a sperm cell is penetrating the wall of an egg cell, producing a zygote. If pre-zygotic mechanisms of reproductive isolation are present, then this stage will not ever happen. In the right-hand picture, the egg is fertilized and successfully becomes a zygote. However, if post-zygotic mechanisms of reproductive isolation occur, then the zygote could either be aborted preterm or may live but could be sterile and a non-producing member of the population. With pre-zygotic mechanisms, there are two main types: allopatric and sympatric. Allopatric means other place. This is when organisms are separated by a geographical distance and become isolated from other closely related species. Sympatric means same place, and these species live in the same region as closely related organisms, but become isolated based on various factors which reduce the likelihood of interbreeding. It is important to realize that although many of these species do not interact in nature, many are closely related enough that they could produce viable and fertile offspring if they were in a laboratory setting, like a liger. or a zorse, a zebra-horse combo. They won't do it in nature, but we could do it in a force scenario. With allopatric speciation, a physical geographic barrier prevents two species from interacting. After many generations, genetic drift and mutations in each group will prevent successful breeding between the two species. If enough time passes and the geography changes again, which allows the two species to once again mingle, they will be too different genetically to reproduce with each other. Two specific ways that allopatric speciation can occur are through dispersal and vicariance. With dispersal, as the word implies, a small group will leave or disperse from the original population and over time will become a new species through adaptation to the new location. Notice the left side of this diagram. There are two lizards which get washed away to the isolated island. Over time, they create their own population with a unique set of genes as compared to their ancestors. With vicariance, nature causes a physical barrier, such as a flood rerouting a river or an earthquake splitting the land. Notice on the right side of the diagram, a force of nature causes the river to change course, resulting in a split between members of the original population. Over time, there are two different species split by the river. In both cases, once enough time has elapsed, the two populations will be genetically different to the point they can no longer interbreed, producing viable fertile offspring, and as a result, speciation will have occurred. Another type of evolution is when multiple species diverge from one parent species. In the event of the Galapagos finches, as birds radiated out to a new area from the parental population and adapted to their new environment, speciation occurred, creating numerous species of finches. For a fun animated version of allopatric adaptive radiation, watch the linked video. When two similar species living in the same region have different preferences such as food choices, habitat locations, or mating behaviors, it results in sympatric speciation. These choices separate them from other species, thereby reducing the chances of interbreeding. The thin-lipped versus the thick-lipped cichlid species live in the same lake but prefer different food sources, which has resulted in a change in their morphology or body shape. And this change prevents females from selecting males from the other species. Once enough time has passed, the genetics of the two populations will have changed to the point that they will become separate species. Similarly, in the apple maggot flies, the original population preferred the red hawthorns, but when apples were introduced into the same area, some flies began laying eggs on the new host. This difference in host became a reproductive isolator, and now the two fly species cannot interbreed. Watch the linked video on the difference between allopatric and sympatric speciation using these two examples. Another example of sympatric speciation is when two closely related species can breed and produce a zygote, but the resulting embryo does not survive, resulting in miscarriage or stillbirth. Therefore, the hybrid is not possible. Yet another example is when the hybrid offspring will be viable, but could be sterile. Consider the case of crossing a donkey and a horse, which produces a mule. A mule is a very strong, able animal, but is sterile. This lack of fertility happens because the number of chromosomes between these two parental species do not match, and therefore the mule has an odd set, which results in sterility. Post-zygotic hybrid reproductive issues can also occur in viable hybrids where future generations are impacted. This is known as hybrid breakdown. Here is an example of a plant's hybrid breakdown. Two fertile hybrids are able to reproduce, but the resulting F2 generation is weak and not fertile. As Darwin and Wallace studied species around the world, several components of science became increasingly important to support the theory of evolution. Fossils of similar but sometimes extinct species found on different continents, as well as anatomical features which have similar appearances and/or functions, provided necessary evidence for evolution. Organisms will adapt to survive, and over time the population can evolve to sustain the species. A beneficial trait in one environment might be a deadly trait in another environment, so evolution happens as needed and not in a predetermined path, such as a quote, quote, bigger organism is always better and beneficial. That's not always true. One individual will not evolve, but may hold traits that increase their chances of survival and therefore reproduction. When a group of individuals pass on specific traits, these traits could become more common in the population and therefore shift the genetic frequency. Over many generations, one species may evolve into two or more species. So we have made it to the end of chapter 11. This is the end of all the chapter material for this course, Biology 1408. Make sure you go back and review the end of the chapter questions for both chapters 10 and 11. There could possibly be some bonus questions from previous chapters, but they will only be bonus questions. So this test, your final, will be on August the 6th, which kind of gives you about a 4 or 5 day period of no assignments, or about a week, I guess, actually. And then, so this last week, make sure you complete your discussion post. You've got that plus your 2 responses to get full credit. That needs to be done before 11:59 on Thursday. And this week you only have an extra credit lab. There's no regular 4 credit lab. If you want to do extra credit, that's great. If not, you don't have to. But if you do the extra credit, it has to be turned in no later than 11:59 p.m. on Thursday as well for any credit. If it's turned in after that, no credit will be awarded. If you have any questions, as always, email me, and I hope you have a great week.